Good evening, everybody. I'm just going to allow people to get into the room here for a bit, and then we'll get started. Well, welcome to the last One Health seminar of the spring semester. Hope you've been with us through much of the semester. We've got another great presentation for you tonight. Before I get into kind of a discussion of what One Health is and the introduction of our guest tonight, I just want to first say happy day of scholarship to all the Del students and faculty and staff who may be on this particular Zoom. And also want to congratulate all of you students and faculty and staff who were recognized at the Founders Day ceremony this afternoon. You should be very proud of your accomplishments, and we're very proud to have you at Del Val. Well, for those of you who may not be familiar with One Health, and I would suspect that probably all of you are quite familiar with the concept of One Health, very simplistically, it's just a recognition that we have one planet, and we got one future together. And so, if we ignore the environment, if we ignore the plants and animals that we share this planet with, it'll be to our peril. Our kind of definition that we have down at the bottom of the slide, One Health is the transdisciplinary systems approach. Well, it's recognizing the planet as a single system. And transdisciplinary is pretty simple. Just need to listen to each other. We need to step across lines of disciplines and different backgrounds and talk to each other. That dealing with problems in a transdisciplinary approach, boy, I tell you, for you students who are getting ready to start your careers, if you learn this lesson early, you're going to be way ahead of the game. Being able to actually go and step out of your discipline and say, listen, we've got a problem here, and I think that maybe you might be able to help with it. I think you'll find that the better solutions will come from that kind of activity. Here at Del Val, we've been working with this kind of broad concept of that everybody at Del Val ought to be aware of One Health and how it might fit into their lives. For my students, they're quite aware that when I say, what is One Health about? The answer I'm anticipating is everything. It's about everything. Three basic ways of approaching it. If I can get my slide to go ahead, is through education. We have our students in their first year are introduced to the concept of One Health in Del Val Experience 1 or 2, I'm sorry. We also have it embedded into a large number of courses, and certainly anyone who's had a course with me has heard considerable about One Health and how we should be paying attention to these other elements of the triad besides ourselves, or if we're in animal-related majors, besides the animals as well. Research as well as working with others to come up with solutions to particular problems. And then finally, outreach, looking at getting the message out and encouraging others to recognize that we're all on this planet together and that our future is linked, all of us, to each other. So with that, I would like to introduce our speaker for this evening. Let me stop my sharing. And I'll allow Dr. Abdelaziz to bring up his presentation. And let me just read from our flyer here, his introduction. Very happy to have Khaled with us this evening. He earned both his master's degree and his PhD from Beni Suef University in Egypt before completing a postdoctoral training in the Department of Pathobiology at the Ontario Veterinary College, University of Guelph. Following this, he served as a research associate in the same department. Prior to joining Clemson University, Dr. Abelaziz gained valuable industry experience in the field of poultry vaccines. He worked at Seva Animal Health Incorporated in Canada as an innovation and development scientist, contributing to the development and licensing of vaccines against poultry coccidiosis. Currently, Dr. Abelaziz holds the position of assistant professor of immunology and microbiology at the College of Agriculture, Forestry, and Life Sciences at Clemson University. He is also a faculty scholar at Clemson University School of Health Research. His current onehealth research at Clemson is focused on developing non-antibiotic solutions, including vaccines and probiotics to effectively control foodborne pathogens in chickens. Thank you so much for being with us this evening, Khaled. I really appreciate you coming forward with your research, which I think we're going to find very, very interesting.
Thank you, Dr. Hyatt, for the introduction. Good evening, everyone. Thank you for the introduction and for allowing me this opportunity to share our One Health approach to control foodborne pathogens in the chickens with the ultimate goal to prevent its transmission to humans through contaminated poultry products. And thank you all for joining my webinar at this late hour of the day. So our current research is focused on controlling foodborne pathogens, particularly Campylobacter in chickens, as I mentioned, to prevent its transmission to humans. But why Campylobacter? Because according to the Center for Disease Prevention and Control, Campylobacter is considered the #1 cause of bacterial diarrheal illness in the United States. And according to the Public Health Agency of Canada, Campylobacter is the main cause of gastrointestinal illness. Between 2011 and 2015, there were around 10,000 cases of Campylobacteriosis were reported annually in Canada. And here in the US, as we can see, there has been a drastic increase in the number of confirmed cases of Campylobacteriosis through FoodNet. These numbers represent a 13% increase in the incidence of infection, Campylobacter infection, compared to data from 2006, and an approximate 70% increase compared to data from 2016. And a recent study conducted in Europe indicated that the number of Campylobacter cases is expected to increase in the coming years due to climate change. The healthcare costs incurred by this, bacteria. One second. The healthcare costs incurred by this bacteria in the US were estimated to be between $2 and $6 billion. So Campylobacter species are motile gram-negative bacteria. They normally inhabit, the gut of the domestic animals, including chickens, and close to half of all bacterial foodborne infections are caused by Campylobacter, which makes this pathogen the leading bacterial cause of human gastroenteritis in the world. Campylobacter now surpassed salmonellosis. In a recent report released by CDC, there were around 1.3 million cases reported annually for Campylobacteriosis compared to only 800,000 cases of salmonellosis. Environmental contamination and farm animals are sources of Campylobacter infection. It is estimated that over 90% of all human cases of Campylobacteriosis originate from farm animals, among which chickens are considered a major source of infection. And while Campylobacter infection in farm animals is not associated with any clinical signs, its transmission to human through contaminated animal products, especially poultry products, can cause acute enteritis, and only 500 bacteria can make humans sick. Campylobacteriosis in healthy individuals is a self-limiting disease. However, Campylobacter infection in children, elderly, and immune-compromised individuals can lead to severe clinical manifestations that require antimicrobial treatment. Symptoms are ranged from mild to bloody diarrhea, abdominal pain, vomiting, and in severe cases, gastrointestinal perforation, irritable bowel syndrome, and Guillain-Barré syndrome. So as we can see here in this table, the prevalence of Campylobacter in retail chicken meat. And the prevalence rate here varies from one country to another. And there is an inconsistency in the prevalence rate in these countries. And this could be attributed to regional differences, such as differences in farming practices, differences in food safety regulations, and other environmental factors. It could also be attributed to differences in the sampling methods, detection methods, and surveillance systems. So Campylobacter causes persistent infection in farm animals with little or no inflammation and essentially no clinical signs. However, in humans, it causes severe intestinal inflammation. It's unclear whether this distinction due to differences in the structure and the function of the immune system of animals and humans, or due to the ability of Campylobacter to evade or subvert the human immune system. So the major goal of combating infection in farm animals is to reduce intestinal colonization and in humans to prevent clinical disease. However, no commercial Campylobacter vaccines are currently licensed for, or products are currently licensed for humans and animals. Let me pause here. I'm going to hide this. Okay, that's good. So in addition to environmental contamination and farm animals as sources of Campylobacter infection, fresh vegetables, fruits, and environmental water sources were reported as risk factor of Campylobacter infection. So this triangular dependency on human health, animal health, and their environment is an important basis for controlling this disease. So to facilitate the implementation of a One Health approach against human campylobacteriosis, and expertise from veterinarians, human medical experts, policymakers, industry and farmers, public health authorities, researchers, and regulatory agencies are needed or should be included in future concepts for controlling Campylobacter. We really need to control Campylobacter in farm animals, particularly chickens, to prevent its dissemination to the environment and transmission to humans. We have recently published this review article that summarize the mitigation strategies to control Campylobacter at different stages of the food chain, from the stable to the table. And this includes pre-harvest control, post-harvest control, and public health awareness. At the pre-harvest level, several intervention strategies have been applied to reduce Campylobacter colonization rates in poultry farms, including biosecurity measures, dietary manipulation, antimicrobial alternatives, genetic selection, and vaccination. However, these strategies have been only partially effective in reducing Campylobacter burden in poultry flocks. So at the post-harvest level, two methods are being used in slaughter plants to decontaminate the chicken carcasses, including physical method and chemical methods. For physical methods includes hot water followed by crust freezing, and the combination of ultrasound and steam treatment. And for chemical methods include detergents, various acids, and chlorine formulation. In addition to these methods, biological methods such as the use of essential oils, bacteriophages, probiotics, and others to reduce the Campylobacter load on poultry carcasses are currently under investigation. So it's really important to raise public health awareness about proper handling of raw poultry meat and the importance of kitchen hygiene to minimize the risk of infection. Dr. Hyatt, can you play the video from your side, please? Dr. Hyatt? You are muted, Dr. Hyde, you're muted. Hello? Okay.
There, I'm
Yeah,
back again. I
yeah.
just had it muted. Let me go back here and start that again. So, Khaled, were you seeing the image, or do I need to have you get out of share and I'll
Okay.
share this?
Okay. Yeah.
All right, let me go ahead and I'll share my screen.
Okay. Yeah.
And hopefully this is correct. There. Are you seeing that?
No. Don't be. So I put it here in the chat box. So for you, so I don't know, the sound doesn't actually work from here. So I put it in the chat box. I will try actually to explain what's going on. Okay, let me share my video here.
So sorry, I thought that
Oh,
we
no
would
problem.
be able to see it from there.
Okay, that's fine. Do you see my presentation now?
Yes. Can
Okay.
see your presentation. We can
Okay. Yeah.
see the video, but not hearing anything. And also, you might go up to your display setting and just say duplicate the setting so that we're only
Okay,
seeing your single slide. Yep. Now we lost.
Yeah, that was good.
Now we lost your
Okay, you lost my
presentation.
I'm sorry.
There you go. And
Okay.
so get back to your slide again.
Okay, now we see it.
There you go.
Okay.
No sound, but we're seeing it.
Okay. So now they spray the chicken with this fluorescent material.
Yeah, we're seeing that.
Yeah. And they use the UV light here to see the carcass itself is heavily contaminated with this fluorescent material. Look at the knives, his hands. Yeah, and then he touches his face here. So let's splash the bug around. Oh, yeah, and onto the chicken that was also clean. Yeah, so the piece of advice here is not to wash the chicken actually in the sink because this spreads the bug around. So raising the awareness about this, the public health awareness about the importance of kitchen hygiene would actually result in minimizing the transmission to humans. So we're currently employing a One Health approach to mitigate Campylobacter infection in humans through feeding chick embryos and hatchet chicks with probiotics. But what are probiotics and why are they promising targets for controlling Campylobacter infection in poultry? Probiotics are defined as live microorganisms that, when administered in adequate amount, they confer various health benefits to the host, including the modulation of the gut immune system and the gut microbiome composition, and production of enzymes and organic acids that play an important role in digestion and metabolism, in addition to their immunoregulatory activities or immunomodulatory activities. Probiotics can also improve, gut barrier function and competitively exclude the microorganisms from the gut, especially pathogenic microorganisms, by adhering to the attachment sites on the small intestine and also compete these microorganisms for nutrients. So we have recently assessed the immunomodulatory and anti-Campylobacter activities of probiotic lactobacilli in vitro, and we found them to exhibit differential antagonistic effects or inhibitory effects against Campylobacter. Here we employed two approaches, radial diffusion, and as you can see, the zone of inhibition here, and also the co-culture or killing assay, and as we can see, there is reduction in colony forming units in the groups that were or the Campylobacter that was co-cultured with probiotics. We also demonstrated that these probiotics abrogate the quorum sensing signal in Campylobacter. But what is the quorum sensing signal? It's a process of cell-to-cell communication that allows bacteria to share information about cell density and adjust their gene expression accordingly. As we can see here, there was a significant reduction in the quorum sensing, or what's called the auto-inducer protein. Probiotic lactobacilli also reduced the expression of virulence genes in Campylobacter, including genes responsible for Campylobacter motility, genes responsible for adhesion, and genes responsible for invasion, a gene responsible for cytotoxin production, and the LOX-S gene that's responsible for quorum sensing. Lactobacilli also enhanced macrophage phagocytosis of Campylobacter and reduced the Campylobacter invasion of human intestinal epithelial cells. We also found that lactobacilli enhanced the expression of cytokines in chicken macrophages, including Th1 type cytokine, interferon gamma, interleukin 12, interleukin 1 beta, and CXCL2, which is a chemokine, and interleukin 10, immunoregulatory cytokine. We also found that lactobacilli enhances the expression of macrophages for macrophage co-stimulatory molecules or surface markers, including CD80, CD86, and CD40. So these results actually encouraged us to investigate whether manipulation of the gut microbiota of chick embryos and hatchet chicks with probiotics will enhance chicks' resistance to Campylobacter infection. And to investigate this, we explored several questions. The first question here is, will in ovo administration of probiotics affect the hatchability rate? The second question, will probiotics accelerate the development of lymphoid organs? And the third question, will they modulate the gut immune responses? The fourth question, will they modulate the microbiome composition? And the last question here, will probiotics enhance chicks' resistance to Campylobacter infection? And this is the site of injection. Before we answer these questions, I'm gonna play this video to show you how we inoculate the fertilized eggs with probiotics. Here, we need here to turn off the light to candle the egg. As we can see, this dark body here is the chick embryo. The orange fluid here, let me go back, pause. The dark body here is the chick embryo. The orange fluid is the amniotic fluid. And here, this is the air sac. So the next step is to make a circle around the yolk sac to determine where exactly we're gonna inject the probiotics. Now he will turn on the light and he will mark the center of this circle. That's where exactly we insert the needle for for probiotic administration. So he marked it. We spray the eggs with ethanol to prevent the introduction of any pathogenic microorganisms from the eggshell into the chick embryo. And then he will use this piercing device to make a hole in the eggshell. Okay, now he made the hole. So in the next step, he will inject the probiotics. So here we use 13 gauge needle and as you see, the insulin syringe for injection. Then he will go all the way down to the amniotic fluid. So if we feel any resistance there, this means we are hitting the embryo. So we go back a little bit and we inject the probiotics. Now he's injecting it. And then we need to seal this hole. We use Premont or waxing material to seal the hole. You just apply it, to the hole here and leave it for a couple of minutes. Then we place, the eggs back into the incubator. So we usually vaccinate or treat, the eggs at embryonic day 18. So one might ask, what if these probiotics are capable of achieving these effects? Is this a feasible route for mass administration and commercial poultry production? So let's watch this video and I will answer this question. That's what happens in commercial poultry production. So as we watched, the vaccinators are being used in commercial poultry production for mass vaccinations of thousands of eggs, and this actually is very suitable and a feasible route as opposed to the post-hatch laborious procedures. And this route actually is being used here in the US and worldwide to vaccinate chickens or chicks against various viral diseases such as infectious bursal disease or gumboro and Marexi disease, with over 90% of commercial poultry production here in the US using this route for vaccinating eggs against or chick embryos against Marexi disease virus. Okay, so back to the first question. Will in ovo administration of probiotics affect the hatchability rate? And we found that when we treat, these chick embryos with probiotics, it improved the hatchability rate by 2.3%. While this percentage may seem modest, it holds significant importance in production terms. So that's great. It didn't negatively affect the hatchability rate. It improved it. So the second question is, will probiotics accelerate the development of lymphoid organs? We found that, in ovo injection of these probiotics, differentially alters the expression of genes involved in the development and immunological maturation of bursa of fabricius, which is a major lymphoid organ that's responsible for antibody production in chickens. They alter the expression, as we can see. There was higher expression of interferon gamma, interleukin 12, interleukin 17, transforming growth factor, B cell activating factor, and B cell activating receptors. So this has led to the conclusion that in ovo delivery of lactobacilli provides signal not only for B and T cell development, but also for promoting their immune function and survival during bursal development. To the third question, will probiotics modulate the microbiome composition of hatchy chicks? Here we divided fertilized eggs into 4 groups. One group received probiotics in ovo only, no post-hatch administration of probiotics. The second group received probiotics at day 1 post-hatch, followed by weekly administration until week 5 post-hatch. And the third group received it, in ovo and post-hatch until week 5. And the fourth group here did not receive any probiotics and served as a negative control group. Then we analyzed the alpha diversity, which is a measure of species richness, which means the number of taxa, and species also evenness, which is the relative abundances of those taxa. And our results indicated that the group received probiotics in ovo had higher richness and evenness compared to the control group, but this was only observed at the second week post-hatch. No differences were observed at third week, fourth week, and the fifth week of age. With regards to alpha diversity, and there's actually a measure of variability in community composition or the similarities and dissimilarities among the treated groups, we found that beta diversity analysis revealed a distinct separation among the treatment groups, with probiotic treated groups displayed diversification compared to the control group that is circled by this green circle here. As you can see this for week 1, first week, and second week as well. For the third week, we can see a partial overlap in the composition there. And for the fourth week, there is an incomplete overlap. We noticed a complete overlap by week 5. And let me see here, this could be because of in poultry or chickens, the microbiome stabilizes between the second week and third week. So it seems like those probiotics had an effect on the microbiome composition until the second and third week somehow, but not the fourth and fifth week. And these results align with the taxonomic composition of the cecal microbiota. So let's discuss it week by week. As we can see here, there was a lower there was a lower abundance of proteobacteria, phylum proteobacteria, compared to the control group in the orally treated birds, in the in ovo treated, and the combination of oral and in ovo treated. And by the way, proteobacteria includes members of pathogenic bacteria of food safety concerns, including Campylobacter, Salmonella, E. coli, Vibrio, Yersinia, and many other pathogenic bacteria. And as we can see here, for this one, there was cyanobacteria, the phylum cyanobacteria. We can see also the abundance was lowered or reduced by probiotic treatment. It was significantly reduced here. There was also a significant reduction, and we can see it here for the orally treated one. On the other hand, we can see the increase in the abundance of some beneficial phyla, including phyla chloroflexi and planktomycetes, which both play a role in host metabolism. In addition to that, planktomycetes can also produce some small molecules such as bacteriocins that have antimicrobial activity against pathogenic microorganisms. And in the second week, we see consistent reduction in the abundance of phylum proteobacteria, and also there was higher abundance here in the phylum planktomycetes and chloroflexi. By the third week, we see consistent reduction only in the groups that was treated with in ovo and orally with probiotics. However, no differences in the other treated groups. And as we can see here, there is no major differences or significant differences in the abundance of planktomycetes and cyanobacteria and chloroflexi as well. By the fourth week here, we can see how the microbiome stabilizes, and we don't see any major effects of the probiotics on the microbiome composition. And by the fifth week, there was no differences among the treated and non-treated groups in terms of the taxonomic composition. So, the fourth question: will probiotics modulate gut immune responses of chick embryos and hatched chicks? When we inoculated these probiotics, in ovo, and delivered them orally, to chicks, we found that irrespective of the treatment, the probiotics reduced the expression of pro-inflammatory, genes, including interleukin 8, which is a chemokine, interleukin 1 beta, interleukin 6, and interferon gamma. And this triggered us to ask a question. Can we use these probiotics to prevent and treat colibacillosis and salmonellosis in young chicks? This is actually a very interesting topic that warrants investigation, given their immunoregulatory activity. So what we propose to do next is to increase the frequency of probiotic administration from weekly, and instead of giving just one dose weekly, to give it daily, to encapsulate those probiotics and apply them to the drinking water or the feed, to the birds, so that they can get them on daily basis. That we also propose to examine their protective role against E. coli, Salmonella, and Campylobacter in an experimental challenge model. We are also interested in investigating whether co-administration of prebiotics would reinforce or enhance the probiotic effectiveness. And by the way, the blend of prebiotics and probiotics is called synbiotic. But what are prebiotics? These are non-digestible carbohydrates that act as a fuel source for probiotics. They act as food for probiotics despite being indigestible by the host. And evidence suggests that feeding chickens these prebiotics enhance the growth of probiotics in the chicken gut. In addition to enhancing the growth of probiotics, prebiotic fermentation by probiotics lead to also production of short-chain fatty acids such as butyrate, lactate, acetate, and propionate, which possess several beneficial effects, including improving intestinal barrier integrity. They also can serve as energy source or food, for enterocytes. They also contribute to elimination of gut pathogens, and this actually was based on some in vitro and in vivo studies because of their bactericidal activity. And more importantly, they also possess anti-inflammatory activity and therefore can be used to restrain excessive inflammatory responses induced by enteric pathogens. What does this mean? It means they can, if we feed them during the time of infection or with pathogenic microorganisms that cause enteritis, such as Clostridium perfringens, they may counteract the inflammatory effect of the Clostridium perfringens. In addition to that, the short chain fatty acids can also lower the pH of the gut, making the condition unfavorable for the growth of pathogenic bacteria. So here we propose to use lentils-derived prebiotics because of the interesting findings obtained recently from feeding those prebiotics to mice, and it was found that they increase the abundance, modulate the gut microbiota by increasing the abundance of beneficial bacteria, including firmicutes and bacteroidetes, which are indicators of a healthy gut microbiota, and lower the abundance of proteobacteria that actually encompasses many pathogenic microorganisms of food safety concern. Soon we will have a symbiotic feed additives that can be used for improving poultry gut health and controlling foodborne pathogens in chickens. So in summary, probiotic lactobacilli can enhance the hatchability rate of fertilized chicken eggs. They can also improve poultry gut health by modulating the gut microbiome composition by enhancing the relative abundance of beneficial bacteria and reducing the abundance of harmful bacteria such as proteobacteria. They were also found they could also serve as therapeutic agents for treating gastroenteritis in chickens and potentially in humans given their immunoregulatory effects. However, further research is needed to explore the role in controlling colonization with foodborne pathogens an experimental challenge model with or without, the prebiotics. So we have another line of research that focus on developing microbial ecosystem therapeutics or next generation probiotics for treatment of Campylobacter-induced intestinal damage in humans. So although fluoroquinolones have proven successful in treating cases of human Campylobacteriosis, the emergence and dissemination of fluoroquinolone-resistant Campylobacter have limited the success of fluoroquinolones in treating this disease in humans. Therefore, in 2005, fluoroquinolones were banned for use in poultry production by FDA, and despite these measures, the antibiotic withdrawal from poultry production did not reduce the antimicrobial resistance rates. Therefore, there is an urgent need for effective alternatives to antibiotics to treat human Campylobacteriosis and reduce or mitigate antimicrobial resistance responsible for like 700,000 annual deaths globally. And gut microbiota has become a promising target for therapeutic interventions due to its wide impact in the host, including gut metabolic and immune system homeostasis, improving the gut barrier function, and the role in protecting the gut against enteric pathogens. A previous study in murine model showed that probiotic supplementation ameliorated the intestinal inflammation caused by Campylobacter by enhancing the production of interleukin-10, which is a key immunoregulatory cytokine. Another study in a porcine model showed that oral administration of probiotic lactobacilli also ameliorated salmonella-induced inflammation or enteritis through enhancing the expression of interleukin 22, which plays a crucial role in repairing the epithelial cells post-injury. While much interest has focused on the use of single-species probiotics to mitigate intestinal inflammation, their effect tend to be transient and limited. Therefore, attention is now turning to the use of more complex microbial communities that engraft within the gastrointestinal tract, and such interest has been driven by the recent success of fecal microbial transplant in treating gut dysbiosis and enteric diseases caused by Clostridium difficile, E. coli, and Campylobacter jejuni. And the good news here is FDA allows the FMT only for treatment of Clostridium difficile cases that are resistant to standard antibiotic therapy. However, it should be noted that fecal microbial transplant lacks batch-to-batch consistency, and they may carry over many undesirable microorganisms from the donor like individual to the recipient or the patient. So, due to safety concerns and the lack of proper standardization and safety of fecal microbial transplant, a well-defined, consistent, and safer microbiome therapeutics are warranted. For that, we are currently developing microbial ecosystem therapeutics for treating Campylobacter-induced intestinal damage and restoring the dysbiotic gut microbiome using pigs as an experimental model of human Campylobacteriosis. But why pigs? Because the anatomical and functional similarities of the pig and human intestine, the digestive physiology, intestinal digestive enzymes, and secretory proteins of pigs are quite similar to humans. The mucosal and intestinal B and T lymphocyte population in pigs are quite similar to humans, and more importantly, pigs can be used to replicate, the gut microbiota of human through fecal microbial transplant. So we hypothesize that, oral administration or supplementation of community probiotics that possess anti-inflammatory and tissue repairing properties to Campylobacter infected pigs will lead to increased production of anti-inflammatory cytokines such as interleukin-10, transforming growth factor beta, and interleukin-35, and will also increase the production of cytokines that are implicated in tissue regeneration and repair, such as interleukin-22 and interleukin-23, leading to amelioration of gut inflammation. We also hypothesize that if we provide, the infected pigs or sick pigs with these, community probiotics, they will modulate, the perturbed gut microbiota, leading to recovery of a healthy gut microbiota. We believe that this project is innovative because we intend to develop a consortium of beneficial microbial communities by selecting specific bacterial strains based on their anti-campylobacter, anti-inflammatory, and tissue repairing capabilities. And how these, next generation probiotics or microbial consortium differ from the traditional probiotics. So for traditional probiotics, there are only single or a few species that act alone. However, in microbial consortiums, there is microbial synergy. Bugs support each other to create an overall larger benefit. And what's the difference between fecal microbial transplant and our microbial consortium? Our microbial consortium copies, the fecal microbial transplant. However, it will be well-defined, well-characterized, acceptable, stable, and safer. And on top of this, it will be targeted against specific pathogen. So in summary, the alarming rise in the emergence and the spread of antimicrobial resistance and the associated global impact necessitates an urgent intervention of alternatives to combat, the growing threat of antibiotic resistant bacteria. And with the poultry industry heading for a post-antibiotic area, or like no antibiotic ever, antimicrobial alternatives are needed to fill this gap, needed to compensate for antibiotic effects. Probiotics have been shown to be promising targets for replacing antibiotics in poultry feed. We can use those probiotics to manage foodborne pathogens in poultry to prevent their transmission to humans and reduce the healthcare costs incurred by these pathogens. Research should transition from relying solely on the traditional probiotics, which typically consist of a single or a few species, to embracing next generation probiotics, and future research should prioritize employing a holistic One Health approach to ensure the safety of animals, humans, and the environment. So I would like to acknowledge, the funding agencies NIH, South Carolina Department of Agriculture, by South Carolina Biocraft Center, and the Clemson University for supporting our research. And I would like also to thank my team for their help and contributing to these studies. And in the end, I would like to thank you all for joining my webinar at this late hour of the day, and I would be happy to answer your questions.
Thank you so much, Khaled. Folks, if you'd like to put your questions into the Q&A, we can go ahead with that. We have a couple of questions from Phoebe. I think maybe you've already answered these, but I'll bring them up anyway. I assume your egg that is inoculated with probiotics is incubated, and you did show incubators for that. And then secondly, how would inoculation be accomplished in the field? Collect the eggs at what stage of development, incubate them and inoculate, then hatch the chicks? Will this be practical? Seems like it's quite mechanized, isn't it?
Yeah, that's a good question. So usually what we do manually, as I showed in the video, we do it manually by marking, the center of, the air cell, and then make a hole, then introduce those probiotics into eggs. And in the other video, I showed that, we have now some vaccinators, so we can use those vaccinators to inject, the eggs with probiotics. Usually the injection time is 18 days of incubation. Okay, and the eggs hatch at day 21. So following injection, we place these eggs back into the incubator until the hatching day, which is day 21. Then, the chicks hatch, and then we put them in the poultry barn.
So I have a question for you,
Yeah,
Khaled. So we earlier in the semester, we actually had a presentation, or maybe it was last semester, we had a presentation related to One Health certification in the poultry industry. So do you see, and certainly they are moving away from any kind of antibiotics.
yeah.
Do you see this particular technique being potentially incorporated as part of the One Health certification for the poultry industry?
Of course. Yeah, so absolutely. I see that, it's very promising actually, like, a technique to be included in the One Health certification because now in poultry production with the ban of use and of antibiotics since 2017, actually, we are like the producers are striving to find alternatives to antibiotics. So we are exploring like, several antimicrobial alternatives everywhere worldwide, including, the probiotics, prebiotics, bacteriophages, essential oils, organic acids, enzymes, etc., to compensate, for the antibiotic effect. To fill this gap, that's actually like at the level of the poultry production in humans as well. The emergence of antimicrobial resistance requires us to find alternatives to antibiotics. Okay, and now we're using some probiotics that you can find them in yogurt. You can eat them in yogurt. You can find some probiotics on like and on shelves in pharmacies and grocery stores. But we need actually those probiotics to be targeted against a specific pathogen. We need those probiotics actually to restore the gut or reshape the gut microbiota, okay, following infection and even after antibiotic treatments. So if we resort to use antibiotics, we need to reshape or reset the gut microbiota. So we rely on probiotics in doing this. And there is a product that is, actually has been released for Clostridium perfringens. It's called New Biota. So New Biota is actually a product that was produced by a group in Canada that founded a company called New Biota Company, if I can recall the name correctly, and this was found to be successful in treating cases of Clostridium difficile. And as you can see in my research, I'm trying also to develop something similar. Instead of using fecal microbial transplants, we don't know what's inside. Maybe if we use fecal microbial transplants to treat one disease, we may introduce another pathogen. So we treat one disease and introduce another disease to the patient. So we need to work on improving the probiotics. We need to move from using traditional like probiotics to next generation probiotics. So adding this to the certification, I found it to be very beneficial, and I'm here to help with that.
Great, great. Anonymous has a question. This
Yeah.
research seems monumental, however, may be hard to implement in the field. Has there been any research in applying this concept a step back in feed for the hens, in feed for the chicks?
Yes, there are many commercial like probiotics that are available, like as a dietary supplement for feed. But our group actually is one of the first groups to investigate in ovo inoculation of probiotics. And the point of that, we need to introduce those probiotics to developing that to see the developing gut, like the gut gastrointestinal tract. We need to those probiotics to attach to like the adhesion sites in the small intestine and close the door for any pathogenic microorganisms that may actually like adhere to those sites post hatch. During this time actually is very critical because the chickens are vulnerable to many diseases, like especially the first week or second week post hatch. So we need to introduce them early on time to modulate the gut microbiota, to see the gut of like chicken embryos so that they have their beneficial like probiotics in their gut after hatching. We need also to modulate the gut immune system early on time. We need these probiotics to accelerate the development of lymphoid organs. And we are currently working on, because of our research and interesting findings that we obtained recently, that in ovo injection of those probiotics accelerated the development of bursa of fabricius, which is a major lymphoid organ for antibody production. We are thinking of in ovo vaccinating those eggs, and also co-administering the probiotics to enhance the development of bursa of fabricius, so that we can increase the antibody titer specific to the injected vaccine. In field, yes, there are many commercial probiotics, but none of those probiotics are targeted against Campylobacter or Salmonella or E. coli. There are there to improve the poultry gut health. They are there to improve the growth performance of the birds. And that's actually the unique part of our research, is when we develop them, we need them to also be targeted against specific pathogens. And the feasibility, I see it's a visible route, because we're going to use those vaccinators actually to use the same technology to introduce those probiotics to the chick embryos.
And Anonymous had another question, which is, would this process be viable to inoculate against other diseases, or would multiple injections be antagonistic?
No, there is no antagonism, because this method is being used here in the commercial production for vaccination against some viral diseases like Gumboro or infectious bursal disease and Merck's disease virus, and we don't see any antagonism there.
Great. We have time for maybe one more question, if folks have something burning they would like to ask. I think your research is amazing, and certainly looks like it's going to have some incredible effects, not only on the poultry industry, but also will have far-reaching impacts for human health as well. And certainly we've seen in the human society, I'm old enough that we've seen an increase in people's health issues related to having problems with dust or with other things, in which, for us old people, we always said you had to eat some dirt so that you would get your immune system really boosted. Certainly for those people who are suffering from intestinal issues, this certainly looks like it has some hope for improving that situation.
Yeah, hopefully. And this actually, the point of our work is to introduce those probiotics to the gut. So because we need to the lymphoid organs within the gastrointestinal tract to be exposed to the antigen, and we need this antigen to be beneficial antigen. So that's a point. That's why we see some improvement in the development of bursa of fabricius there.
And I think that Anonymous just said, how do you see this making the jump to mammalian medicine? I think you've addressed that certainly that is the point, is to jump to mammalian medicine as well, particularly us.
Yeah, yeah. We still, I'm a veterinarian, and I'm using an animal model for that. So at this point, we are using pigs. But when I get to the clinical status, of course, I will collaborate with human medical expert on that. So, but this area of research, I'm combating Campylobacter, as you see, in farm animals and also in humans. And this is actually the One Health approach.
Perfect. Well, we are out of time. Thank you so much for a great presentation. Thank you to all of you who have stuck with us through the semester. I wish everybody a great end of the semester, and hope you have a wonderful evening. Good night, everybody.
Thank you again, Dr. Hyatt, and hope you have a wonderful evening, everyone. Okay, bye-bye.
Thank you. Good night.
Thank you. Bye-bye.